It has no eyes, no brain, no muscle in its legs, and it walks. Not drifts, not floats, walks in herds across the deepest, darkest floor on Earth. Scotoplanes globosa, the sea pig, pale pink, bloated, the size of a potato. Hjalmar Thael first described it in 1882, working from specimens dragged up by the HMS Challenger expedition between 1872 and 1876. He noted something that stopped him cold. The tube feet, he wrote, are modified into elongate processes that elevate the body above the sediment surface, not gripping, not clinging, elevated, moving. These animals walk on hydraulics. Each leg is a hollow cylinder, pressurized by an internal water vascular system. No muscles, just water pressure, extending, contracting, extending again. And they do not walk alone. Rogachiva and colleagues publishing in Marine Biodiversity in 2013 called Scotoplanes globosa one of the most abundant megafaunal species on the abyssal plains of the world ocean. In some regions, sea cucumbers account for more than 90% of all megafaunal biomass. That figure comes from Billett and colleagues, published in Oceanography and Marine Biology Annual Review in 2001. 90%. The deep ocean floor is not empty. It belongs to them. Which raises the question nobody has cleanly answered. The abyssal plain is featureless, cold, lightless, kilometers of gray-brown sediment in every direction, undisturbed for centuries. How does a blind, brainless animal find food? An animal with only a primitive nerve ring in place of anything resembling a brain. How does it navigate? How do hundreds of them end up moving in the same direction at the same time? That question has a specific answer, a logged answer, a filmed answer. By the end of this video, one ROV dive number and the footage it captured will reframe everything you think you know about how these animals move through the dark. The mechanism science still cannot fully map. The simple answer seemed obvious. Sea pigs eat sediment. Sediment blankets the entire abyssal plain. So they drift wherever currents take them, graze whatever falls beneath them, and ask nothing more of the ocean floor. For decades, that explanation held. Marine snow, the constant slow rain of dead plankton, fecal pellets, and organic debris, drifts downward at 1 centimeter per second. It settles evenly across thousands of square kilometers of gray mud. If your food is everywhere, you need no navigation, you just stop where you land. Andrew Thurber's 2012 research at Oregon State confirmed the feeding biology. Sea pigs process sediment at extraordinary rates, extracting bacteria and organic particles from mud that would look like nothing to any other animal. The abyssal plain is their buffet. Every square meter, the same. The distribution data seemed to confirm it. Rogachiva's 2013 taxonomic survey, published in Marine Biodiversity, found Scotoplanes globosa scattered across ocean basins in patterns consistent with passive bottom water dispersal, slow currents, slow animals, slow accumulation, nothing to explain beyond basic oceanography. Researchers drew the arrow on the whiteboard. Current flows this direction. Animals end up in that region. Case closed. Except the numbers from one specific dive didn't fit the arrow. In 2002, an MBARI research vessel deployed an ROV to 1,674 meters depth in the Pacific. Dive log T257. What the camera recorded stopped the analysis cold. Over 300 individual sea pigs converging on a single point from multiple compass directions simultaneously. All bodies oriented the same way, moving inward, not drifting, not grazing randomly, moving with clear directionality toward one location. Passive current drift cannot produce that. Currents don't flow inward from every direction at once toward a single coordinate. Something was calling them, from kilometers away, through total darkness. With no eyes, no brain, no centralized sensory structure science has fully mapped. If the current wasn't doing the steering, what was? Here is the part that actually changes everything. Sea pigs can smell, not with a nose, not with a centralized organ science has fully mapped, with their entire body. The surface of Scotoplanes globosa is studded with microscopic papillae, thousands of them distributed across the body wall and feeding tentacles, each one a potential chemosensory site, each one reading the water. According to the Holothurian chemosensory literature, including the foundational deep-sea work by Billett and colleagues in 2001, these animals detect dissolved sulfides and amino acids released by decomposing organic matter. The chemistry of death drifting upward through the sediment column. No centralized brain processes this signal. Only a diffuse nerve net spreads across the body wall with no headquarters, no command center. The signal arrives everywhere at once. That should be enough to explain what they do. It is not. In July 1998, Craig Smith at the University of Hawaii deployed an artificial whale fall experiment in the deep Pacific. Sea cucumbers colonized within weeks, hundreds of them, oriented, moving with purpose. Smith's 2008 comprehensive study documented this pattern repeatedly, aggregations arriving not randomly, not by accident, but directed. Here is the problem. Passive chemical diffusion has limits. In abyssal bottom currents, dissolved compounds stretch into narrow filaments, not expanding spheres. Andrew Thurber, a researcher at Oregon State, calculated feeding rate data in 2012 that implied animals were responding from distances far beyond what diffusion chemistry alone can reliably carry. The MBARI T257 dive log recorded animals converging from estimated distances exceeding 1 kilometer. 1 kilometer of pitch black, near-freezing sea floor with no eyes, no brain, no GPS. Diffusion chemistry explains what happens when they arrive. It does not explain how they knew to start moving in the first place. Something is extending the range. The literature has not named it yet. Dive T257, Monterey Bay Aquarium Research Institute, 2002. The ROV descended to 1,674 meters, and what the camera recorded is still cited in scientific literature 23 years later. The log shows over 300 individual Scotoplanes globosa converging on a single gray whale carcass, not scattered, not random, oriented, moving in formation from multiple bearings, all pointed toward the same source. 300 animals, one carcass, one convergence point in absolute darkness. Craig Smith and Anabako documented the event in the 2003 Oceanography and Marine Biology Annual Review. The published paper flagged something specific. The approach distances exceeded what passive diffusion could explain given the recorded bottom current velocities at that site. The animals arrived from farther away than the current alone could have carried the scent. No subsequent study has closed that gap. That is the receipt. That is what Chapter 1 was pointing toward. An animal with no eyes, no centralized brain, no sensory organ science has fully described, navigating kilometers of featureless sediment with enough precision to arrive at the same exact coordinate as 300 of its kind, from different directions, but simultaneously. Billett and colleagues calculated that sea cucumbers can comprise more than 90% of total megafaunal biomass in some abyssal regions, the most abundant large animal on the abyssal plain, covering more sea floor by biomass than almost anything else alive down there. And it finds food by a mechanism science cannot fully map. This is not a gap at the edges of knowledge. This is a gap at the center of it, in the most common creature on the deepest floor on Earth, still walking, still converging, still arriving pointed the right direction every single time. The ocean is 11 kilometers deep in places. Most of it has never been filmed. T257 gave us one dive, one carcass, one convergence, and the question it raised has no answer yet. If you found this fascinating, subscribe. We investigate strange, real creatures like this every single day.